Welcome everyone to Blooducation Baristas, where we're serving you piping hot transfusion news and now conversations on advancing medicine topics. My name is Daniela Hermelin, and I'm the Chief Medical Officer of Impact Life Blood Center and Assistant Professor of Pathology at St. Louis University School of Medicine. And today we're going to be discussing a really interesting topic. Seems that I talk a lot about hemolytic disease of the fetus and newborn these days, but I couldn't help myself to bring in two expert speakers, one a patient advocate, Bethany Withersby she's actually the director of Allo Hope and herself a true patient advocate, and also Dr. Ken Moisey, who is the co-director of the maternal fetal medicine facility at UT Health in Austin. And so today we're going to be speaking about a new drug that is right now in the clinical 3-phase trial known as Nipocalimab. This is a monoclonal antibody that can possibly be used to treat early stages of hemolytic disease of the fetus and newborn. So I'm excited to share this information with you to see, how it can be used, maybe even discuss everything from how the mechanism of action to talking with Bethany to understand, where this could really help the patient and hear her perspective on that. So I'd love to get started by bringing in my friend Bethany to speak about, Allo Hope, this incredible nonprofit organization that she started, and to get her perspective on this new drug. Welcome, Bethany. So nice to have you.
Hi, thanks. I'm excited to be here, and this is a fun topic, actually, for me. So very excited.
Well, it's definitely a new type of therapy. That's it's so incredible that we're at a stage where we've been talking about Rh immunoglobulin for so many years. I remember it was just about 5 years ago that it was the 50th anniversary of Rh immunoglobulin. And now we're starting to see maybe some new treatments. But before we get started, can you tell us a little bit about yourself? Tell us
Sure.
who you are. I'd love to hear a little bit about your story of Allo Hope, and you're doing some phenomenal things. I'm going to share with you in just a second why I say that.
Oh, wow. Thank you. Okay. Well, I'm Bethany Weathersby, and I'm the founder and executive director of the Allo Hope Foundation. But most importantly, I am an allo-immunized mother myself. I have anti-Kel antibodies and have had 4 severely affected pregnancies. And in my first sensitized pregnancy, my daughter Lucy was stillborn halfway through the pregnancy. And we were told we couldn't have any more living children. And we were obviously devastated. And with the right care and with a different team of doctors out of state, we went on to have 3 healthy Kel positive babies, and they're totally healthy today. They're thriving. And so throughout that experience of growing our family and going through these high risk pregnancies, I started a blog to just chronicle our journey. And women from all over the world started reaching out to me with questions about their own allo-immunized pregnancies. And they were telling me that the basic information on my blog that I had provided was the only disease information they were able to find online. And so I realized, oh, wow, this problem is a lot bigger than just my own pregnancies. This is a worldwide situation, and babies are dying. And that really kind of was the spark that ended up being the Allo Hope Foundation. And so here we are today, and we are a nonprofit organization. We're based here in the US, but we provide support and resources to patients worldwide who are facing allo-immunization and HDFN, hemolytic disease of the fetus and newborn. And that's the basic story. That's my condensed story.
Well, thank you for sharing with me. I still hear the pain from Lucy, but you put that into such an incredible, legacy for her. I'm myself emotional just thinking about it. What was that period of time from the blog to the, actually developing the nonprofit?
Mm-hmm. Well, it was really difficult because I was going through these pregnancies myself, and we had been told by our original team of MFMs that um, our babies would die before viability. It was impossible to have a healthy baby with severe disease like I had. And so that was just, always in the back of my mind as we went on to have 3 more pregnancies and face this disease again and again, which is what a lot of these women have to do if they want to grow their family, because once you develop these red cell antibodies, you have them for the rest of your life. And often pregnancies get progressively more severe as you go on. So that's just, we were dealing with that. And then also just understanding that so many patients are struggling to find the right care, to, get to the doctor who can actually provide that care. And also just knowing their baby is at risk and there's very little that they can do about it. It's very, very difficult. So that was just hard, just walking through pregnancies with these other women and doing my best to help, but realizing I'm just one person and we need a bigger platform. And so also throughout that, Dr. Moise was instrumental in starting the foundation. He was my doctor with my pregnancy after Lucy. And so he's the doctor I traveled to get the right care. And it's because of him that we have our 3 youngest living healthy children today. And also he taught me so much about this condition. He's always willing to answer my questions. And actually, back in 2015, he started telling me about this new possible treatment, and he and I would talk for really long periods of time and just kind of fantasize about the future of what if this actually became the treatment? What if this was how we could actually keep babies safe and protect mothers from this psychosocial burden of this disease? So we've been talking about it for almost a decade. Yeah.
Wow. I mean, it's just incredible to hear, what you've done from a blog to a nonprofit, and to understand that you've been helping so many women, so that you could reach a woman, you could tell your story and you can help others with your story. Because I do agree with you. This disease is really hard to understand.
Yeah.
And then to find the right, just the right way to get treatment, the right type of physicians. I can't wait to bring Ken on, but I just want to quickly share with you that just this week, at a medical director meeting, a medical director shared that they recently watched a podcast or a session that you were holding, and they have formulated a new letter to provide patients. And what you're doing is really working. And
That's
so this is a hospital, very big hospital in Illinois, who is now implementing a new type of process to collaborate with maternal fetal medicine in developing letter that is shared. And so it's taking off, Bethany. Your work is really actually leading to some big things. So with that, and with our colleague and mentor, we'd love to bring Ken on. And now kind of talk about this. And thank you so much for all that you do and being such an incredible physician advocate for your patients. Thanks for joining us.
Good morning. How are you? Yeah, I might add that not only she reached out to most of the United States, but now that she's moving into Africa, sub-Saharan Africa. So they're just branching out to the whole world with this message.
Yeah.
Pretty incredible.
Amazing. Well, we came today to speak about that drug that's a clinical in stage 3 clinical trials, I believe. But maybe you can help give us a little bit of background of what this is, and a little bit about how it's been studied so far.
Yeah, thanks for that, Danielle. So basically, Nipocalimab is a monoclonal antibody. It's a humanized, we call it a humanized monoclonal antibody. It's not made in humans, it's made in cell lines, but it looks like other human antibodies. So your body doesn't react to it. And it was engineered to block a specific receptor in the placenta called the neonatal FC receptor. And that's the receptor that allows mom's antibodies to cross over to the baby. Now, normally that's a good thing because you want a baby to gain all the information of immunity from its pregnant mother so that when it's born into the world, it can fight off its own infections before it makes its own antibody. But in cases like Bethany's, bad antibody crosses too that causes, attaches to red blood cells in the baby and causes anemia. So Nipocalimab blocks all the antibodies, not just the bad antibody. It also blocks the good antibodies from crossing the placenta. So it's non-specific. And that's its major mechanism, is its blockade at the placental surface. We know the antibodies start crossing about 12 weeks of gestation, which is, the end of the 3rd month. And then it will continue. The antibody will continue to cross, actually in greater and greater amounts as the pregnancy progresses, to where at term, when the baby's born, the levels in the baby are about 125% of the mother's. So the baby's ready to go when it's born with a lot of immunity that it acquired from mom. And so Nipocalimab is going to block the placenta. The other thing it does, which is fairly unique, it also lowers mom's antibody level in her bloodstream. It works through sort of the same mechanism of this neonatal FC blockade. That receptor lines your blood vessels and allows you to maintain a level of what we call IgG, a specific type of antibody, in your bloodstream. And so by blocking the FC receptor at the placenta, it also blocks it on the blood vessels. And therefore, mom's titer, or the amount of antibody in her bloodstream, will go down by as much as 85%. So we have 2 hits, or 2 powerful mechanisms to help protect the baby.
That is fascinating. Maybe if we could just pull it back even a little bit further. So you're saying that there's an actual receptor on the placenta, and then subsequently you said also on the endothelial vessels. And this receptor acts like a little shuttle. It's like a little ferry that brings across IgG antibody across placenta and into the baby. And the mechanism of action is that this is a monoclonal antibody that blocks, it's an inhibitor antibody that blocks the ferrying, the shuttling of this antibody. But you mentioned it's going to do it, it's not specific, it's going to do it for every antibody. Well, maybe IgG of a certain isotype, IgG antibody. And with that, we're hoping that it blocks, the toxic antibody, the antibody that might be trying to attack the red cell, the protein on the red cell. I think the second thing that you described is a little bit harder for me to wrap my head around. Are you saying that there is a shuttle also, that FcRn is also shuttled on the endothelial cell and is pulling antibody into the blood vessel?
So,
or it just kind of binds?
Yeah, this is a little bit harder. I didn't learn this in immunology school and when I was in medical school. So those Fc receptors are right under the endothelial cell membrane, and they grab the IgG in your bloodstream and sort of protect it from being recycled. So what the Fc is, sort of think of it as a scavenger that protects that IgG. So it gets pulled into the endothelial cell, and instead of being broken down, it returns it back to the circulation. So that Fc receptor, FcRn receptor, sort of scavenges and protects your circulating level of IgG in your bloodstream. So if you give Nipocalimab and you block it, it sits on that Fc receptor, it can't do its job anymore. So what happens is the IgG comes into the cell, and then it's degraded inside the cell. What happens is the maternal blood level of IgG then falls. So there's 2 mechanisms here, but this is one that's not well described, in that you get these circulating levels of IgG that fall because of the blockade.
Do they get degraded, or they just are attached to the receptor?
No, so the receptor has the Nipocalimab sitting on it, and so it can't do its job. So if it didn't have the Nipocalimab, it would grab the normal IgG, it would protect it as it goes through the cycle in the cell, and then they would basically dump it back into the circulation. So the fact that we block the receptor means it can't do its job, and the IgG gets into the cell and gets degraded. And as a result, the levels fall inside the maternal bloodstream.
Got it. So it has essentially two effects. It's going to inhibit the transcytosis, the fancy word for bringing in antibody into the placenta that will end up in the fetal bloodstream. And it will also decrease maternal levels of IgG.
That's correct.
Okay. Now, I think we should focus on the first part, which is what this, is intending to really help us. Is tell us how you would use this drug, and at what stages, and how does this even look right now to you?
So the drug is an intravenous preparation. It's given intravenously in a dose based on mom's weight, and it's given every week. So it wears off. This blockade wears off within a week if you don't keep giving the drug. And so it's given as an infusion that takes about 30 minutes to go into mom's bloodstream. And it hangs around for about a week. So every week there's another infusion that has to be given. It can be started, at least in the current clinical trial, as early as 13 weeks. And then it's given every week up to 35 weeks. Now, pregnancy, as you may know, goes to 40 weeks. That's the due date is 40 weeks. So you might say, well, why are you stopping the drug? We want to allow some of the good antibody to cross at the end of the pregnancy. And for the benefit of the baby, but not allow too much of the bad antibody to cross. So a decision was made to stop it a few weeks before delivery. So we would stop it at 35 weeks and maybe deliver at 37 weeks. So give those last 2 weeks a little chance to get some of the beneficial antibody to cross and not give enough time for the bad antibody to cross in any great amount. So that's how it's used currently.
And I have a question you may not know the answer to, but is there some type of upregulation of that FcRn receptor if you block it? Is there anything such as that? Do you know?
Yeah, that's a great question. I'm not sure I know the answer to that. We don't have a way to measure the FC receptor's response on the placenta. Obviously, we need to leave the placenta intact. And so what we measure is FC receptors on a specific type of white blood cell called a monocyte. It has FC receptors on it also. And we can do an assay to see the blockade that occurs on those cells as a surrogate for placental blockade. At least in the studies so far done, we get 100% blockade on the mononuclear white cells, assuming that means we have a pretty good blockade on the placenta as well. But we're unable to tell if we block all the receptors or whether there is an upregulation that occurs in response to the drug, at least not on the monocytes. We don't know about the placenta.
Mm-hmm. And you don't use that as a pre-screening tool for patients. It's more on the level of just understanding the mechanism of action for research.
That's correct. It was set, that assay was set up to decide from a pharmacokinetics point of view, when does that blockade wear off? And it seems to be about a week. And what dose do you have to give to keep the blockade on board? So that's a surrogate again for placental blockade. But it was used in developing the pharmacokinetics for the trials.
Got it. Well, with Bethany's experience, can you share with us, what type of alloimmunization treatments you experienced, and a little bit about what maybe traditionally exists, and,
Sure.
and how your experience with that?
Yes. Well, it can really differ between countries. So we're talking about, the US here, and I also have a very extremely severe case. And so most women do not have this level of disease as I do. But for me, it was, a lot of blood tests, a lot of ultrasound, so weekly MCA Doppler scans. With my 3 living babies, we started those at 15 weeks. So that's every week, lots of appointments. And then I had to do plasmapheresis and IVIG to delay the onset of fetal anemia, to try to, get the baby to a gestation where it's safer to do the IUT, the intrauterine transfusion. And so then I had consistent intrauterine blood transfusions until delivery. And then there was the post-birth care of, phototherapy for hyperbilirubinemia, for the baby, and then top-up transfusions. And they were finished with the disease at about 15 weeks old.
Wow.
Yeah.
So
So
you mentioned IVIG, you mentioned plasma exchange, you mentioned blood tests, you mentioned ultrasounds, you mentioned phototherapy. What I didn't hear you mention is all the anxiety and worry throughout that whole time.
Absolutely. Yeah. Thank you for bringing that up. We can often just focus on the baby because the baby is the one at physical risk, but the mother does carry a lot herself. And this is a rare condition, and it requires complex, time-sensitive management. That means, many healthcare providers just don't have enough experience treating the disease to be well versed in the management of it. And so patients end up having to kind of take ownership over their own care and then advocate for the right care often. And so that's a lot. That's a lot. And I had to do that with mine, especially I tried my best in my first sensitized pregnancy. And I wasn't able to get the right care, which is what led to my daughter's death. And so for me, I had to travel out of state. It was an 11-hour drive to get to Dr. Moise. And so I had to relocate and live in the Ronald McDonald House for 5 months. And with my two youngest babies, I also had to travel out of state. And that's a lot. That is a lot. And so mothers with this condition are at a very high risk for anxiety, depression, PTSD, and just feeling very isolated. And that's how I felt, for sure.
I'm sorry. But I imagine that with what you're doing, you're creating a network of parents that can help support each other and physicians that can give them the support they need as well. So
yeah,
thank you for that.
And I will say, having a doctor like Dr. Moise, having them who does provide the right care, makes such a huge difference for the mother and the baby. So I'm thankful for MFMs like Dr. Moise.
Danielle, might I add one thing? I've also experienced when you and so it's not their fault. It's a natural process to develop this immunity to foreign red cells. But in essence, although they're not sick and they have a normal baby, it's normally formed, it doesn't have a birth defect or anything, it just happens to be the wrong blood type, they're rejecting their baby. Their antibodies are hurting their baby. And sort of, I'd love to hear Bethany's comment on this, but it's sort of, they're helpless. They can't change that. They can seek good medical care, but they can't change their antibody. It's there to stay for the rest of their life, and it's going to harm their baby if their baby's the wrong blood type.
Yeah. Yes. Thank you for bringing that up. We struggle with guilt, even though it's completely out of our control, and we would do whatever it takes to keep our babies safe. But it is very difficult to just accept that my body is both creating this life and, intricately weaving together this little human, and also at the same time trying to destroy my baby. My immune system is trying to destroy my baby. And yes, there's very little we can control in that situation. And so it feels very panicky as a mother. I'm sure you can imagine. And so,
yeah,
yes.
I was just going to say, that you're right. It's such a contradiction of, creation and destruction happening in one vessel.
Yes.
I can't help always to philosophize over this. I do believe that the body, was created in like this perfect form, and I'm still trying to figure out the good of why this happens. I think maybe we will understand it. Of course, in your position, you're saying there's nothing good about this, but I would like to understand why the body had evolved to create antibodies that direct against red cell antigen. It's something, I'm sorry to intellectualize it. It is a fascinating idea. Of course, there is an emotional consequence to this, and And it's nice to share that if other women and other parents are feeling that way.
Yeah.
Yeah. Sorry, go ahead.
Well, I just wanted, my take on it is that our bodies are not perfect. There can be all kinds of things going haywire. One little thing can be off, which can create a huge problem. And so, yeah, I wish that our bodies were perfect and functioned in the way that they were originally designed. And unfortunately, sometimes they don't. And so that's my perspective on it. But I do think you can use something terrible like this for good. And you can use your suffering, your pain, and your trauma to help others and bring beauty into the world. And we can carry on our baby's legacy in that way by helping others and protecting other families from this condition.
Absolutely. And perhaps even just understanding the disease with each year and with each scientist and each doctor and each patient advocate, here we are talking about Nipocalimab.
Yeah.
And so, I'm sure, Bethany, you would love to hear, I would love to hear, what are some of the clinical trials that have already been published, and where are we right now with our, I think you mentioned stage 3 clinical trial. Can you share some of that with us?
Yeah, so the development of a drug, particularly to be approved by the FDA, takes 4 steps. And most people may not be, your listeners may not be totally familiar with that. First is preclinical studies. These are typically done in animal models. In this case, they were done in monkeys to see if the drug is safe in an animal that's close to a human. And this was done in monkeys. They actually studied their offspring to be sure that they were okay, that the antibodies that didn't cross weren't going to cause any ill effects. And a lot of studies were done first in these monkeys. Later, what's called a phase 1 trial was done in normal human volunteers. And so they were given the drug at different doses and a lot of blood work done to be sure there weren't any kind of side effects, dangers. And that all went well. And then what we recently published just in early August of this year was called a phase 2 trial. Now, in a phase 2 trial, you do 2 things. You try to figure out how much drug to give, and you also see if there is some effect, and you usually pick the worst case scenarios. So in the trial we just completed and published, we picked patients who had lost a baby, much like Bethany, who had lost a baby early or had a blood transfusion in the womb before 24 weeks. So we call that early onset disease. So in the phase 2 trial, we picked the worst of the worst to see if the drug would work and how much drug we should give. And like I said, we just published that trial. We can go over the results in a minute. Now we're starting the phase 3 trial. So in a phase 3 trial, which is sort of the final trial before drug approval, you have to, again, pick a bigger population of patients with the disease, but you have to do a placebo arm. And the patients sometimes have difficulty understanding that. But basically, the trial gives a drug to certain patients, and other patients get, if you would, sugar water or salt water. And we're not told, we're blinded, they call it. The investigators don't know, the patients don't know. And that takes away any bias of the drug working when we think it is working, because we don't know what we're getting or what we're giving to the patient. So that trial just started. The phase 3 trial just started. I think there's been 7 patients enrolled of 120 patients that we plan to enroll worldwide. There'll be 30 centers around the world that will be enrolling patients currently in that trial. So we're on phase 3. We have to finish that trial, look at the data, and then we'll report that to the FDA and what its equivalent in Europe called the EMA before the drug would be approved and available for widespread use.
Okay, thank you for taking us through those clinical trial phases. I think I always have to review that myself. So you're saying phase 1, human volunteers, it's really helping us to establish safety in a small patient population. So you've checked that box, safety. Okay, now you've done your phase 2, which is looking at more, again, safety, but also a little bit more on efficacy and how much do we actually dose this drug. Can you tell us a little bit about what that looks like again? who were those patients involved in that study, and what were the primary and secondary outcomes for that study?
Yeah, so we picked patients again that had the disease but had really awful outcomes in their previous pregnancy, knowing that if we had an effect, it would be pretty dramatic. Because as Bethany pointed out earlier, in general, the disease gets worse with each subsequent pregnancy. So all of these women, the 13 women in the trial, had a qualifying pregnancy where they either lost a baby before 6 months of pregnancy, or their baby had to have a transfusion very early before 6 months because it was already anemic. And they were alloimmunized either D or KEL. And basically, they were now pregnant again in this next pregnancy, which was obviously destined to have problems. We checked all of them with a test called free fetal DNA, which we've talked about before in this podcast, to make sure their babies, in fact, were positive. They had the antigen involved, so we knew they were at risk. And then they were started on the weekly infusions of the Nipocalimab all the way up to the end of the pregnancy. The primary endpoint was how many patients can we get to 32 weeks without a transfusion? No need for intrauterine transfusion. That was the primary endpoint. Because we figured by 32 weeks, if you get that far without a transfusion, you're going to have a surviving baby. That baby may have some prematurity issues, but it's going to survive in the nursery. And then there were a lot of secondary endpoints like, well, how many transfusions did you need? How many live babies were born? Did fetal hydrops develop? So we looked at a lot of other typical endpoints in HDFN, but we reached the primary endpoint in more than half of the patients, which meant these were women who were destined to have very sick babies. And more than half of the 13 actually got past 32 weeks and never needed a transfusion at all, which was a pretty impressive endpoint considering their history.
Wow. Guys, my belly is a little bit quivering right now. I don't know if you know this. I'm a mother of 6. I've had 6, thank God, healthy pregnancies. But the thought of doing clinical trials on pregnant women makes me so nervous. We just really protect women and their babies. How do we do this? How do you approach this in this ethical way? And which women are signing up to be part of these trials? Can you I would love both, Bethany, your opinion on this as actually a woman who's alloimmunized, who's faced this, because you are a candidate.
Yes.
You are a candidate for this. So I'd love to hear both of your thoughts on this.
Well, I want Bethany to tell the story of the fact that she tried to sign up for the trial with me. I'll let her tell the story.
You want me to do that now?
Yeah.
Okay. Well, yeah, like I said before, Dr. Moise and I were talking about this since 2015, and I turned out to be kind of the perfect candidate for the phase 2 trial because I had a history of severe early disease. And so we'd always wanted a big family, and we thought, what if we could participate and I could avoid all of that terrible invasive treatment and have a healthy baby? That would be incredible. We considered it for like 2 years. We discussed the possibility of it. Dr. Moise answered all my questions very patiently. And then we decided, okay, let's do this. Let's go for it. And right here, I just want to point out that this is a very strange position ethically, because, and a lot of moms just have to do this. Like, if we have a history of such severe disease where we've lost a child or had very early intervention, for the most part, we're not just going to get pregnant again and kind of see what happens. We're going to really think through our options and the risk that we're okay with before we dive into another pregnancy. And so here we are, and we have this clinical trial that seems really promising. But then, we have to get pregnant to be in the trial, and you can't actually find out if you qualify until you are pregnant, right? You can't apply for the trial until you are pregnant, and then you will find out if you qualify. But you have to make the decision to get pregnant first. And so it's like, am I going to get pregnant to be in a clinical trial? This is strange. This feels weird. But actually, the reason we would choose a clinical trial is because we think it is the safest choice for our baby. That's why we would do that. And that's how dangerous this disease can be for those of us with severe early onset disease. And so, yeah, we got pregnant, and then we did have a very well laid out backup plan in case I didn't get into the trial for some reason. And in the end, I didn't get into the trial, which is another long story I won't go into. But so we just went to my backup plan with my MFM, Dr. Tom Trevette in Atlanta. And then I went through all of the treatment again, and he had the most intrauterine blood transfusions. And now he's a healthy 4-year-old, and our family is complete. And so in the end, it turned out great. But I think that's why it's so important for patients to have a backup plan. Yes, going into this.
Well, that is a pretty incredible idea. I've never in my life heard about someone who would, do something to get in the clinical trial.
Right.
Yeah, that is interesting, Bethany. But that you had to resort to your backup plan. But thinking about if you did get into the study, what about just being nervous that you selected something that might not work?
Right.
And that, you're taking that risk, but I guess that's what you do.
Well,
and
exactly.
Taking a risk for yourself and somebody else.
Right. The one person in the world that you feel compelled to protect the most, right? And so it's you have to think about first of all, I don't know if Dr. Moise mentioned this or not, but plasmapheresis and IVIG, which people with severe disease often use to delay that fetal anemia, you cannot use those treatments if you're in this trial. You cannot use plasmapheresis and IVIG. So you're taking that off the table if you do participate. And so that does feel a bit risky. But I think it's the idea that this could also prevent the risk of IUT or, further treatment, because those do come with risks themselves, post-birth and prenatal. And so it's a weighing of the benefit versus the risk. And in the end, the only person who can make that decision is the mother, I think. So, yeah. And also, can I just say personally, I just read all of the research on it myself. And so I loved reading the research on the primates and the phase 1 trial and the transplacental transfer. There's an article about that. And then I discussed it with Dr. Moise. And that was extremely helpful for me to see, in that study, it was showing that the drug doesn't go through the placenta to the developing fetus. Correct, Dr. Moise?
That's correct.
Right. So that all of that gave me a sense of safety for me and for my child, if I chose that path.
Yeah, Danielle, might I add, this was an interesting discussion with both the EMA and the FDA, because think about this, the mother's healthy. In all the other clinical trials, the patient has a disease already, and then you provide them maybe an investigational drug to try to make their disease better. Well, these moms are healthy. They only have disease when when a baby's inside, and they really don't have the disease. It's their baby. So to my knowledge, this is the only trial in pregnancy where we were not treating the mother. We were, in essence, treating her baby, her unborn child. And the ethics of that are very interesting. And the FDA pushed back on us a bit about that and said, well, you have transfusions. Why are you doing this trial? And both Bethany and I know that the transfusions, particularly in inexperienced hands, can be very risky to the baby, especially if they're done early. So being able to avoid transfusions or put them off to later in pregnancy makes sense. What we do have, and the reason Johnson & Johnson selected this disease in pregnancy to treat, is that we have salvage therapy. We have transfusions. So even in the current phase 3 trial, the patients get the same standard of care, even if they're in the placebo arm, with ultrasounds every week. And should there be a problem, we can move to transfusions. Now, it's not perfect. We'd rather the drug work. But J&J decided, well, let's do it in favor of the patient. We'll do a 2-to-1 randomization. That means that in 3 patients, 2 will get the drug and 1 will get placebo and probably end up with transfusions. And let's do the transfusions only at experienced centers who can tell us they've had experience dealing with this disease quite a bit. So it's a 2-to-1 randomization, not 1-to-1 like most trials in the phase 3 trial. But the reason, and I remember the discussion about a different disease in pregnancy, why we won the day, was we had the ability with ultrasound to detect a failure, if you would, of the drug, and we could provide transfusions to save that baby. So we weren't just throwing our hands up and saying, I hope the drug works. We had a way to deal with the pregnancy to get the best outcome.
You mentioned that intrauterine transfusions do pose risk. Can you describe why that would be? And are there certain situations, and how high is that risk? Do we know? Published?
Yeah, so there are published data on the risk of intrauterine transfusions. Before 20 weeks, so if you end up with a baby that's anemic before 20 weeks, the loss rate can be as high as 20%, even in the best of hands, because the baby's just too fragile. The vessels are too small to put a needle under ultrasound guidance into the umbilical cord. That's the lifeline of the baby. After 22 weeks, the risk goes down between 5 and 8% in experienced hands. And Bethany knows more stories than anybody about what happens in centers that don't have a lot of experience. And so when I quote a 5 to 8% loss rate, that's in experienced centers. But you don't, and what people need to understand, you don't have 1 or 2 of these, you have 4 to 6 of these procedures. So you can imagine the mother. In our case, we do it in the operating room, going back to the operating room 6 different times with the possibility each time of a complication that might result in an emergency delivery of the baby, because maybe the heart rate slows down during the procedure and we have to take the baby by emergency C-section. So it's quite onersome to the moms to deal with the anxiety of going to the OR so many times, and the possibility each time there can be some complication with the procedure. So if we could do away with these procedures and treat this disease medically, that's a major breakthrough in this disease.
Mm-hmm. It's disgusting of me to bring this up. I hate talking about it, but what about the financial benefits? I was just thinking, every time, a unit is ordered for a neonate for an intrauterine transfusion, so much scheduling, so much testing. And then I didn't even think about the OR needing to be scheduled, and there's just so much involved in a single intrauterine transfusion. And then I thought, wow, what about the cost of one compared to maybe the cost of these infusions?
All right. And we have an anesthesiologist there, and we have several physicians involved. We have a blood bank involved, as you know. So there is a cost to every one of these procedures. I don't know what the drug's going to cost. I would imagine it's not going to be a relatively cheap drug. But I think if we take into effect the fact that we can negate transfusions and take all that cost off the table, and you could receive this drug as an infusion in any city. You wouldn't have to travel, as Bethany did, to an experienced center miles away. You could receive the drug with some type of surveillance at a local hospital and treat the disease. So more women could be treated effectively in their own cities with this medication, if it all plays out the way we want it to.
Yeah, better access to care. And then you mentioned also, you're able to monitor, quote, failure. Can you just clarify what that means? What would it mean to you as a physician to say, I don't think this drug is working, failing?
Well, I'll say based on the phase 2, I think the drug is working. Let's start with that. I think a failure would be that we would lose a baby early in the pregnancy despite the drug. That did happen in one case in the trial. We're still sort of perplexed as to why we lost that one baby. So 12 women took babies home. One did not. There was a complication of a transfusion, and that baby died in utero. So for me, the home run would be you don't need any transfusions, right? That would be just the best. Okay. I think that there will be some patients with very high titers where we don't succeed in no transfusions, but we succeed in getting them so far into the pregnancy that a transfusion becomes quite easy, or we deliver the baby prematurely. And that's a win too. And so that would be another endpoint for the trial that might be worth looking at in very severe cases. But there were severe cases in the phase 2 trial, and there were many patients at the higher doses that didn't get any transfusions at all. So a win would be no transfusions. A secondary win would be late transfusions. And so we'll see. I mean, the reason the phase 3 trial is being done is to prove that efficacy in a larger population.
Mm-hmm. And so maybe we should talk about this phase 3, both about where it's at right now, both physically and just where you're along with the study. And then I'd love for you to maybe go into any of the risks, the adverse risks that we might be seeing or facing with this.
Sure. So phase 3 is planned for 120 patients. It's called a randomized double-blind study. As we mentioned earlier, the double-blind means the investigator doesn't know if the patient's getting the drug and the patient doesn't know. So we're both blinded to the treatment. It's a 2-to-1 randomization to placebo. So 2 patients out of 3 will get the drug, and 1 patient out of 3 will get a salt solution given intravenously, or a placebo. Some people would call it a sugar pill in the case of taking oral meds. And then all the patients will be followed very carefully at these experience centers every week with ultrasound. And should there be a problem with the ultrasound, we, of course, would proceed to a blood sampling of the baby and intrauterine transfusion if needed. And so that's the way the protocol is set up. So there'll be 120 patients in the trial throughout the world. Currently, there are 30 centers open around the world. I think 7 patients have been in the trial. The first patient, I believe, delivered last week. I can share that with you without talking about the outcome. We don't know if she got the drug or not, because we're blinded to that. So the trial is moving along. In the United States, there are now 6 different centers open throughout the United States, mostly in the central part of the country and the East Coast, but we're hoping to open some centers in the West Coast. And Johnson & Johnson is willing to support travel and housing for patients to travel to the centers that are approved to get the proper care. And get the infusions, and then later the patients would deliver at those centers so that the study can be completed. And so, like I said, the trial is ongoing. I believe we've actually talked and tested 14 patients, and half of them had a negative baby, which was good news for them. That meant their pregnancy was going to be uncomplicated, so they didn't enroll in the trial.
Well, can you clarify then what are the eligibility requirements for this study? When would a woman get enrolled ideally? At what week gestation? And what kind of testing would she have to conduct?
Great question. So we're looking for patients that have had at least 1 transfusion in a previous pregnancy or a fetal loss. So one of those 2 criteria. So not someone who had a baby that maybe needed a blood transfusion after birth. They had to have had a transfusion in the womb, an intrauterine transfusion during the pregnancy, or they unfortunately lost a baby from the disease earlier along. That's the initial qualifying situation. They can't have other medical comorbidities either. Most women of reproductive age don't. But then early in the pregnancy, at about 10 to 12 weeks, we can take a blood sample from the mother to see if the baby carries the red cell antigen or not. So the baby has to be antigen positive, and it's for D, Kel, little c, and big E. So one of the major blood group problems that would cause an incompatibility and fetal anemia. So one of the first steps is to get that blood work done to see if the baby in fact is at risk. If it's not at risk, the patient doesn't enroll in the trial. There are other tests to do, blood test on the mother to be sure she's in good health. We do an EKG on the mother. We do an early ultrasound to make sure the baby is okay before the start of the trial. And if they meet all those criteria, then the first infusion starts between 13 and 14 weeks. But we have to get the first one started by 16 completed weeks. So that's one of the limitations of the trials. We have to find out from the patients fairly early that they're pregnant, and we can move them along through this screening process so they qualify for the first infusion before 16 weeks. Why do we pick 16 weeks? We want to be sure we're blocking that antibody early along so we don't end up with a sick baby soon afterwards.
Right. It makes me think of Bethany's experience of trying to be proactive, very proactive about getting enrolled. So it's important that maternal fetal medicine and other obstetricians know about this so that they can be offering it early at this first appointment, possibly.
Yeah, we've sent a letter to all of the maternal fetal specialists in the United States to let them know about the eligibility criteria. I've actually talked to 7 different patients through Zoom calls who are not pregnant but think about getting pregnant or thinking about getting pregnant, like Bethany was talking about, to see if they might qualify for the trial, and let them, discuss the trial and see if that's something they would be interested in if they decide to get pregnant. So we're open to talking to patients who aren't even pregnant yet, to see if they would be interested in the trial should they should they become pregnant at a later point.
You mentioned earlier on when we talked about the mechanism of this drug that it does block all antibody. That means what we say, the healthy antibody that protects us from bacterial, viral infections. And if I recall, there is a little bit of immunosuppressive quality of a pregnant patient. She tends to get more infections, possibly sinus infections. Is there a higher risk of infection with this drug that have you been able to see that?
Yeah, that's a great question. It's obviously one of the major watches or concerns we have with this drug. And there are 2 things that are reassuring. First, in the 13 pregnant patients, we saw routine types of infections. 2 patients actually got COVID because the study was ongoing during COVID. And although, as a general rule, COVID has a tendency to be worse in pregnant women, neither were hospitalized. And they had urinary tract infections and some other minor infections you would expect in pregnancy. So we did not see any glaring danger signals about infection. The other thing that's important is this type of drug has been approved for patients with myasthenia gravis. It's not Nipocalimab, but they're a similar set of drugs that block the FcRn receptor. And these are older patients. Many of them are on steroids for myasthenia gravis. And there do not seem to be any danger signals in those patients to date. And they've been on this drug for some of them for several years now. So we're beginning to see data from other clinical uses of these types of drugs in older patients who are not showing overt signs of infection. The good news is, although it lowers your immunity, it appears you can still respond to an infection. So it only affects the level of IgG, but if, let's say, you were exposed to flu, you can mount a response with IgG. It just doesn't stay around as long. And it doesn't affect any of your other antibodies, all the other classes of antibodies. It doesn't affect your T cells, which are important in immunity also. So it's only IgG that it affects, not the other classes of antibodies.
Right. Well, it's always interesting to think, how can we extrapolate findings from one patient population to another? I might have just glitched out, but I was just going to say it's interesting to try to extrapolate if we can take correlations of one patient population to another. I do think that pregnant females are truly have a very special physiology. So it's going to be interesting to see, what will be. I also think when we think about pregnancy, we also think a lot about IgA and how IgA may be important. It doesn't sound like an HDFN, but maybe just fetal immunology, definitely after birth through the transfer of IgA during breastfeeding. So I'll be interested in following up on this. So maybe we should just put in the chat right now the trial itself. Would that be okay? Bethany, is there any information that those who are listening who may be interested, whether they're physicians or other potential patients or patient advocates, what would you advise?
Yeah, well, kind of like we've already mentioned, it's very important to get all the information to the patient upfront and help them understand, answer any questions that they have, because in the end, it is their decision, and we want them to feel empowered and educated, and to make a well-informed choice for their pregnancy and their baby. And so to patients, ask every question you have. Don't feel shy. Write one out, go in with a list of questions, ask all the questions. You can contact us if you want to connect. And we have a trial participant support group just through the foundation online. If you would like to join that and you're an allo-immunized mother, come find us on Facebook. And our support group on Facebook, just the general one, is called Support for Antibodies in Pregnancy, and Allo Hope patient support group. So that's a great place to get more information, to kind of discuss this with other patients and feel supported in that way. But I think one of the biggest things is to make sure you have a backup plan with your MFM as if you're applying to participate in the trial, you need to have a solid backup plan. I think that's most of it.
I did put the link in the participant chat for the trial and for Allo Hope Foundation. I don't know how to put in for the chat, maybe the Facebook page.
Yeah, they can find, if they find us, the foundation, we can lead them to that. But that's true. The www.azaleatrial.com, that's a good one. That's through Johnson & Johnson.
Okay.
Below.
Okay, great.
Great.
And now I was wondering if we could just one last question to kind of extend it even to a broader idea, which is you guys are going to be going to that conference in November, the Thor conference, where it sounds like an amazing conference regarding just having sessions about the potential of transfusing incompatible units in trauma to females of childbearing age. Day one. Day two is thinking about guidance. How do we provide support, monitor, etc. in those situations? I can't help but to think about, where does this drug potentially come in? Because right now we think about RH immunoglobulin. If there's a situation where you're transfusing RHD incompatible units, at this point, most centers, most transfusion services or emergency rooms, they don't infuse RH immunoglobulin because the amount of incompatible red cells, it would not make sense. But I wonder, have you thought about how you could use this, or does this have a role in even in that conversation? Will this be brought up there?
I think the conference is mainly going to talk about some guidelines for the treatment of the disease. I don't think we're going to spend a lot of time on prophylaxis. Obviously, prophylaxis is the right answer for this disease to prevent you from getting it to start with. But the trial that's scheduled for November is to look at specific guidelines for people who either receive Rh positive.
Ken, by accident, I think you froze for that last comment. Bethany, was that true for you?
Yes, I couldn't hear the last comment.
Yeah, I can repeat it. So the November conference is going to be basically emphasizing what happens if someone is exposed to Rh positive blood, particularly in a trauma situation. What do we do with that patient? That's a new paradigm that's taking place in some trauma centers and EMT centers, where we're using O positive, low titer blood to treat patients in the field. And so we're sort of helping them, particularly with Allo Hope's help, to decide what do you do with that patient if she does become allo-immunized. So that's going to be the emphasis of the trial. I don't think we're going to address prophylaxis to any great extent in the trial. It's mostly going to be related to treatment.
Bethany, do you agree with that it's not really the time and the place to talk about this potential drug?
I get, yeah. I mean, we're focusing on getting some solid guidelines in place for treatment. And I think that once it's closer to being approved and actually an actual treatment choice, I think that's going to be a big discussion, and I look forward to that. And I think it's gonna be wonderful and shift how we treat this condition. But we're not there yet.
Okay, got it. So putting a little bit the cart before the horse on this one. Okay, yeah, amazing. Well, we really did cover a lot. We covered what the drug is, how it works, the current clinical trials, where we are, how we approach this on an ethical level. We talked about it personally, Bethany, you know, how you would approach this if you got involved in a clinical trial with this drug. And we also put in the chat how to access the clinical trial. What did we miss? What else would you guys like to add?
I think we did a pretty good job of covering everything. Sometimes I get asked, do we have any information on the babies? Our oldest baby was the first baby I treated. She's now about 5 years old and she's doing just fine. And so we did follow the babies. The babies get followed for 2 years and they all seem to be doing well. They didn't have any overt infections that we wouldn't expect for a normal baby in the first couple of years of life. So that's encouraging. But all the babies in the phase 3 trial are going to be followed out to 2 years.
Okay, wow. Well, maybe while we're finishing up, if there's anybody listening that has any questions, please feel free to put them in the chat. We'd love to answer any questions you have, but Bethany did kindly offer the ability to connect with her even afterwards. Bethany, what are some of the, if there are any final ideas that you wanted to share that we may have missed?
Well, I just want to say that as a patient, we are incredibly excited about this. Some patients have been asking me for years for updates. When is it going to be available? And again, we have to wait until we are sure that it's safe and that it is effective. But it's just so wonderful to have the medical community kind of turn towards us and all come together for this reason of helping us protect our babies from this condition. So just want to say thank you to everyone who, I know that this is a ton of work being put into this, and thank you to everyone who is working on this new drug. And we're very excited. We can't wait to see what happens in the future. We're very excited.
You know, it's an amazing place and time to be where we are actually having all of these voices at the table. Yes. You know, again, this upcoming conference, the role that you and your organization have played is amazing. I love that this conversation is beyond just physicians, but with patient advocates and patients themselves. We have every single, kind of subspecialty plus other, and that's the way it should be. And thank you for your courage. Thank you for what you've done. Thank you for the support you've given. It's just unbelievable, really. Mm-hmm. So many of us, we thank you for everything that you've done.
Thank you. It's a gift. It feels like a gift and a privilege for me.
Well, I want to thank everyone who has been listening today. I don't see any questions right now, but again, you know how to contact us. I want to thank both Bethany, Ken. Thank you so much for taking the time to share this with everyone. I truly have learned a lot. I'm very excited to read the published studies of the phase 3 trial of Azalea, and I look forward to seeing you in the future.
Thanks for having us.
Thank you.
Thank you. Bye.